Good morning, everyone. Thank you for the organizing committee for selecting this work for this presentation, for these sessions. I am very glad to share with you the global 48-week result from Monday's study based on dual therapy with darunavir, ritonavir plus 3TC, comparing a standard of treatment in people, HIV people living with HIV. I don't have conflict of interest. This was an initiative, investigator initiative study supported by Richmond Pharmaceutical and the Ministry of Science and Technology from Argentina. As you know, dual therapy has been explored in different studies, different settings, and different drug combinations. On the other hand, in Argentina, a generic fixed dose combination of darunavir 800 milligram and ritonavir 100 milligram is available for naive treatment, naive treatment. Therefore, in 2015, we designed a study to compare efficacy and safety of the fixed dose combination plus 3TC, comparing a standard of treatment, a triple therapy based on the same drug plus tenofovir difumarate. The primary endpoint of the study was the percentage of patients with viral load less than 50 copies in an intention to treat exposed analysis at week 48. This was a randomized, open-label, phase 4 study, which included 336 participants in two stages, as per planned protocol. The main inclusion criteria are described in the box. The randomization was performed in two arms, dual therapy, when we include 171 subjects, and triple therapy, when we include 165 subjects. Of note, this study, the first stage, the result of the first stage of this study was presented in 2018 at CROI, when we include 145 participants for the analysis, and here and today we are presenting the pool analysis of both stages. Regarding baseline characteristics, as you can see in this slide, distribution between age, gender, risk behavior, CD4 count, viral load, and CDC stage were similar between arms. Overall, they had a good CD4 status, and most of them were asymptomatic at the enrollment. Of note, about 20% of the participants had a plasma viral load over 100,000 copies. In terms of the results, the primary endpoint of the study, in an intention to treat the exposed analysis using FDA snapshot algorithm, showed non-inferiority between arms with 93% in triple therapy and 91% in dual therapy. Also, we observed the same results, similar result, the non-inferiority in per protocol population. When we analyze the population with more than 100,000 copies, we found similar response, but it failed to demonstrate non-inferiority because the limit of the margin is exceed the 12% planned by protocol. But of note also that this analysis was not pre-planned by protocol. 7 patients had protocol-defined virological failure through week 48. At failure, the viral load was low, and in the patient when the virus could be amplified, we didn't find resistant mutations. Regarding immunological response or reconstitution, we can see that the recovery of the CD4 cell was similar between arms, but an increase of 2,200 cells in both arms, over 2,000. In relation to safety profile, we observed about 20% of the subjects with adverse event grade 2 or higher in the arm triple therapy and 12% in dual therapy with a little difference between arms. Only few adverse events, grade 2 or higher, were reported in more than 1% of the participants, being the rash the most frequent, followed by diarrhea and abdominal pain that were more frequent in the triple therapy. Adverse events leading to discontinuation were unusual and similar between arms. Although 20 serious adverse events occurred during the study, only 2 had a possible relation to study treatment, both of them in the triple therapy arm. Following the abnormality in laboratory, we didn't find difference between arms or toxicity in the comparison of both arms. But when we compare the values, the change of the values between baseline and week 48, we find a difference on the lipid profile, being higher in the dual therapy. However, these changes, these values do not exceed the normal values, as we can see in the slide. In conclusion, a generic fixed-dose combination of darunavir, ritonavir plus 3TC showed non-inferiority compared to a standard of treatment in the both analysis, intention to treat and per protocol population. Virological failure was observed at low level in both treatment arms. Dual therapy strategy was safe and well tolerated, and based on the results, we think that this strategy could be considered a possible strategy for treatment naive patient, at least for patients with plasma viral load below 100,000 copies. I would like to thanks to everyone who has contributed to the success of this study, principally the participants and their family, the clinical investigator and their staff. Many of them are here today, and the team of Fundación Huésped for support this study, and principally to Dr. Khan for giving me the opportunity to conduct and present this study today. Thank you for your attention.